My name's Andy Bewinter. I'm a Lyman Professor of Biology and a colleague of George's here at Harvard in the Museum of Comparative Zoology. So it's my honor and privilege to introduce George tonight. Before I do, I wanted to thank the donors that, along with the MCZ and the Department of Organismic and Evolutionary Biology, that made the renovation of the new Fishes Gallery possible. And there will be a reception following George's presentation, a members-only reception. If you're not a member of the HM&H and you'd like to be, and you'd like to go to the reception, see the new gallery space, there's a membership sign up over on the right-hand side here, or your left, my right, which you can sign up after the lecture this evening. So the galleries, before I introduce George, the galleries really represent Harvard's legacy and its commitment to its biological collections. Fishes not the least among them. Indeed, as I'm sure George will describe tonight, the success of fishes represents half of the vertebrate diversity that we now know, over 25,000 species and still counting. So he's always quick to tell me how successful fishes are and how important they are relative to any other vertebrate organism. And our hope is that the gallery, in this case, will serve to acquaint you and future generations with the wonderful diversity of fishes. It was inspired to George, myself, and many of us by Professor Carl Lim. Carl Lim, the late professor of Ichthyology and Curator of Ichthyology here at Harvard, really had a provocative and very humorous intellect in the way in which he presented his science and conveyed that to his students, both at the classroom level and those students working with him in the lab or within the collections. And it's really in honor of Carl Lim that the galleries really meant to inspire sort of new understanding, excitement, and fascination with the biology of fishes. So even in an era in which we learn exciting new things about using molecular methods about the evolutionary biology and diversity of animals and plants on the globe that we share with them, it's really the collections that provide a crucial role in grounding us with the animals themselves, the work that we do with them in the laboratories, which George will talk about the work that he's doing in his lab, and the crucial role that they play in bridging our understanding of their evolution with their biology and the habitats that they've successfully exploited in the world around us. So I've known George for quite some time. We date back to having been fellow graduate students here in OEB at Harvard, faculty colleagues at the University of Chicago, and then returned to Harvard as faculty colleagues here. We teach courses together, we co-advise students together, and it's really been a pleasure and honor of having George as a colleague. George is the Henry Bryant Bigelow Professor and Curator of Ichthyology in the Museum of Comparative Zoology, as well as a professor in the Department of Organismic and Evolutionary Biology. His work, his lab conducts work in the biomechanics of fishes and on the evolution of their form and function. His research has spanned from exploring early on how fishes feed to how they propel themselves through the water with their fins and how they control their movements. His current work bridges over with bioengineering and fluid dynamicists. He's one of the few evolutionary biologists that actually does collaborative work with engineers building bio-inspired robots that are going to be able to navigate aquatic environments. And really, George has been doing some very groundbreaking, exciting work in his lab in this area, and I think he'll share some of that exciting work with you, along with covering the wonderful diversity of fishes and the amazing successes in the habitats that they've been able to exploit. So much of this, I should say, was inspired by Carl Lim's mentorship, his enthusiasm, and his wonderful love of the study of biology and the evolution of fishes. And so really that was inspired George as well as many of his students for the work that they do now. So it's with pleasure that I welcome George to the podium to talk about the grand diversity of fishes, form, function, and evolution.
Well, thank you all very much for coming tonight. It's a beautiful day outside, so it's a little warm in here, but I appreciate very much your willingness to step in from the outside and the fresh air to hear about the diversity of fishes and to hear about the new fish gallery that we just opened here in the Museum of Natural History upstairs. And for those of you who won't see it in a few minutes, please do come back, bring your children, bring friends. It's a lot better than it used to be, as you'll see in a few minutes. So it's a great honor for me tonight to present this lecture to you, which I'd like to make something of an overview, not just of the diversity of fishes, although we'll talk about that a little bit too. Also a little bit about current research in fish biology at Harvard. Fish biology has a long history at Harvard, going back to the early days of biology here, and I'll tell you a little bit about the history of that and some of the strange characters associated with the history of fish biology at Harvard, and tell you a little bit about what goes on behind the scenes when study of fishes here with the research collections. And for my career and me personally, this was really motivated and began with the mentorship of Professor Carl Lim. And so we've named, the gallery has really been renovated in his honor. And he was such an inspiration, such a humorist, such an encouraging individual that I think we could all be very lucky to have one professor like that in our careers. He was a tremendous inspiration. Many of his former students are here now. And I hope that the gallery renovation that you see will remind you in some way of all that Carl was able to contribute here at Harvard. Now, I think that Andy alluded to the vast diversity of fishes and how many more fishes there are than mammals and birds, amphibians and reptiles. So it's very convenient for those of us who work on fishes to use that diversity as a weapon to minimize the importance of other groups of vertebrates. We have to be very careful because the plant biologists here and the insect biologists, we really can't hold a candle to the diversity that they study. But at least from the point of view of fishes, I think, and vertebrates, there's a tremendous diversity of shape, of color, of size. We go from very small fishes here. This is a sexually mature adult you can see on a human fingerprint. Of course, large fishes or pictures like this are all over the web. And now the fish show on one of the television channels, the monster fish or whatever it's called, in search of really big fish. There's a tremendous size variation. This is actually the smallest known adult vertebrate. Fishes have tremendous variation in color, beautiful finned iridescent patterns, beautiful coloration, strange shapes. This is actually a fish. This is a fish, though it may not always look like it. And given the 30,000 species of fishes and beautiful shapes and colors, and those of you who have been snorkeling on coral reefs certainly appreciate the diversity of fishes, you would have thought that over the years Harvard would have a particularly beautiful fish gallery to show you when you go into the museum. And if any of you have been there before the last few weeks, you would see the nice fluorescent lighting, the green background, the uniform green background, the horrible looking fishes. Some of them are cracked. This one looks like it spent, I don't even know what it looks like. Weird labels, pebbles on the bottom here with a very sad looking lungfish. It was really kind of pathetic. And I remember this from my undergraduate days. I've been asking around to see if anybody remembers when this old exhibit was installed, and nobody can really remember. It was at least 50 years. So I think we're looking roughly 50 years this fish exhibit was here. I teach a course in biology of fishes. I can't even have undergraduates go through it. Young kids who go visit the museum are lingering over the mollusks and scooting through the fishes because it just looks so horrible. So I hope you'll come back and see the new exhibit because it's much, much better. And I have to thank the Museum of Natural History staff, Jan and Sylvie in particular, for all the hard work they've done. There are nice pieces of actually scientific information about fish. There's information about the diversity of fish. There's information about the evolution of fish, key features of fish ecology, migration of fish. In that case, it's the tuna. We present information on a number of the classic stories of fish migration around the world's oceans. I'll say a few words about that later on. And as we, after talking about some of the history of fish research here, I will touch on some of the themes that are echoed in the exhibit upstairs. When you have a chance to go up and look at it, you'll recognize that. So there's been a complete renovation with new lighting and new case renovations. The old historic cases are still there, but they look new. Now, the gallery is named in honor of Professor Carl Lim, whom you see here, who was a curator of fishes and professor from 1972 to 2009. And he played such an instrumental role in modernizing fish research here at Harvard. Here you see him teaching in the fish class, pointing out the articulation of the jaw and the skull of a fish. down in the collection. He was just a tremendously influential figure. When he gave his last lecture, we presented him with a cake. He always began the course asking, "What is a fish?" And so we prepared a cake based on that. And his humor and his lecturing style, those of you in the audience who knew him certainly can appreciate that. Those of you who didn't, I just hope you've had someone in your academic lives from kindergarten on up who was so influential as Carl has been to so many of us. And so you'll see a panel on the side of the new exhibit that discusses the fish collection at Harvard and Carl's contributions to the exhibit. One of the key things that Carl did as a curator and professor here at Harvard was to greatly expand the fish collection. So you'll see many examples in the new gallery, but behind the scenes there's a very large collection of biodiversity collection of fishes that's very actively used in research, and many of the other departments here in the museum have similar kinds of collections. In the case of fish, they're typically housed in jars with alcohol to preserve the fish, and are taken out then for study. We have many visitors through to these collections. The collections now are in excellent shape. You'll see some pictures in a moment, but it wasn't always so. And even as recently as 1973, here are some pictures of what the collection looked like. This is roughly around the time of my undergraduate days. I don't want to date myself any worse than that. But if you looked in the fish collection, this is what you would see. Bottles on wooden drawers. I particularly like the wagon that was used to haul the specimens around the collection. It was pretty sad. And Carl undertook a first big renovation of the collection, and in the last 10 years we've been doing a new second renovation to bring it up to modern standards. And so we have specimens where you could find them. They're properly labeled. All the visitors can come through, and we do have many visitors come through and use the collection for research purposes. So this is a very substantial and significant, both historically and in terms of modern specimens, biodiversity collection of fishes. And if we go back into the 60s, we can see we had somewhere around 200,000 specimens, and now we're at almost 1.5 million. And the big growth part of this curve was really due to Carl Lim and his inspiration and effort to modernize the collection and increase the number of specimens. We're running out of space a little bit, so we're tapering off. But now that the renovations are mostly done, I'm encouraged to begin a new acquisitional period for the collection. So there are many specimens here in the collection that are of importance. And some specimens don't look all that great, but they were collected back in 1790. And these are some of the first fish in the collection. They're the Peck specimens. And specimens then were dried. These are from the Piscataqua River in New Hampshire. They were dried, sometimes cut in half, pinned to a board, and stored as a representative of the diversity of fishes that you see. So the collection does have material that goes back to the 1790s. From that time to the present, there have been a series of interesting characters associated with the fish collection. Professor Lim, the most recent Lim era here, was certainly an important character, but Louis Agassiz played a very important role in initiating the collections here in the museum. A number of other important people, Henry Bryant Bigelow, William Schroeder, Giles Mead, were very important. And this character here, Samuel Garman, was quite a strange guy. He worked on sharks primarily. It's said his office was inhabited later by Professor Lim. His office is just a few doors away here. It's said that when he passed away and they went in to clear out his office, that they found a big jar next to his desk that was filled with little pieces of sandwich. So he would have a sandwich for lunch, but he wouldn't finish it, and he would take the bits he hadn't eaten and put it into the jar. And so there was this large jar of sandwich pieces dating for 30, 40 years from his time in the collection. He had other strange personal quirks, but we'll ignore those for the moment. If you search the collection, and although we have 1.4, 1.5 million fish, you can search it all electronically. You can search for some notable collectors who have contributed over the years in various ways. You can search for Henry David Thoreau, the noted American author and naturalist, and you'll find some fishes that Henry David Thoreau collected in Concord. Here they are. This is a fish that was collected in 1859, or a group of fishes. It was donated by Thoreau to the Boston Society of Natural History. which doesn't exist anymore, but in the 60s, those fish that were part of that collection came to Harvard and are now in our collection here. So here's the jar of Thoreau's fishes. There's no precise locality. It's just Concord. We can imagine it was Walden Pond, and that some of that biodiversity from 1859 is present in the collection today. But I want to emphasize, before we move away from collection-related things, the utility of these scientific collections is tremendous, not just for identification of new species, but also doing things that wouldn't maybe occur to you to think were possible, which is sampling tissues of fish collected a long time ago for environmental toxins like mercury, for example. We have fish from the same locality from the early 1800s up to modern times. Mercury doesn't degrade. You can sample the tissues for mercury and look at the history of environmental pollution through time, in addition to the standard and important types of biodiversity research that are done with these collections. We've also had undergraduate students collecting. These are students from my fish course a couple of years ago in the Charles River, just across near the Weld Boathouse. We have permits to do this. You can't just do this on your own, which is a good thing because occasionally the police show up. So this guy's looking, this is me, so he's looking at me, probably wondering where's my permit, but I have a permit. And they were actually very nice and were quite amused to see what we were doing. We sometimes keep a few fish for the collection, but typically everything just goes back into the river. And so this is the modern Harvard undergraduate contribution to fish research here. It was a fun day. I'd like to now to turn to some of the themes that the new exhibit displays and discuss some of them in brief. We have there were many things we could choose from to talk about in the relatively small space that's available in the display. But we have information on evolution and diversity of fishes with some evolutionary histories, such as the one I've shown you here. We have some information about the anatomy of fishes, jaws, particularly the head and the fins and locomotion of fishes. Interesting information on fish migrations, locomotion, and transitioning into the area of robotics, which is a particular interest of mine. But I think more and more interest is taking place in how we can make underwater vehicles better, quieter, more maneuverable than the current ones which use propellers. Propellers are very noisy if you want to observe marine life. Your robot comes in and the propellers are all whirring and you need about 8 propellers to turn those things around. If you saw a Titanic, you know what those robots look like. There are multiple propellers. They're very noisy. And we're thinking that over the next 10 to 20 years, some of the fundamental research on fish locomotion and our robotic research can come together to produce much more biologically realistic or biomimetic fish-like underwater robots. So that's one new direction, and this is a theme which is highlighted in the exhibit upstairs. So let's say a few words about diversity and touch on a few of these themes in the remainder of the talk. So this is the current view of the evolution of fishes, of the grand diversity of fishes. At the base of this evolutionary tree, which starts here, we have the hagfishes and the lampreys, little icons of which are shown here on the right. And there's been some debate over the years as to whether these two groups are joined at a common evolutionary ancestor, or whether they come off separately from the base of the tree. 10 years ago, I would have shown you a diagram where they come off separately. Now these are commonly lumped together. The evidence, I think, is pretty strong that they're the jawless fishes, fishes without jaws. And then everything from this point on up has jaws. We have the sharks and rays, the cartilaginous fishes branching off next. Then the rayfin fishes, which have the vast diversity of fishes. Finally, the coelacanths, a very interesting ancient group with 1, maybe 2 plus living species. There's a representative upstairs in the public galleries. Lungfishes, and then leading to us, which I've represented with a salamander down there. Not to be too human-centric. What I'd like to do is say a few words about some issues related to cartilaginous fish, sharks in particular, and then talk a little bit about rayfin fishes and illustrate some of the themes in the new gallery with information from these two groups, and in particular tell you just a little bit about some of the research that's being done. So if we start with the cartilaginous fishes, there's a tremendous diversity. But there's one unique, or very particularly interesting feature, I should say, one that's interested me about animals such as sharks, and that is the structure of their skin. Now, why would I be interested in shark skin? Seems it's been used to make wallets. It's kind of an obscure academic topic, but there are some interesting things to tell you about this. Sharks are covered with small, bony denticles or scales. And so if you've ever felt the side of a shark or felt shark skin, it feels like sandpaper. And there's quite a diversity of different denticle types depending on where you are on the shark. We'll look at these in more detail in a moment. Very important is to remember or to note that these are bone. So we think of sharks as cartilaginous fishes, that their skeleton is calcified cartilage, but the surface scales are made of bone. And they look like this. Here's an array, an image we've taken in my lab of the shark skin. And the water, when the shark swims, would be going from my left to my right. So water's going to flow over them. They have these sort of lobes with ridges. We can zoom in on one here. They're really quite beautiful structures. Again, they're made of bone, and they're quite small, although there's a lot of variation in sharks. So here's a shark scale. It's about 130 microns. That may not mean much to you. It's about the diameter of a human hair or a thickness of a piece of copy paper. So they can be quite small, and you can see the surface of sharks is paved with these structures. Even more amazing than the fact that they're made out of bone is that each one is like a little tooth. So on the left here, I've taken a textbook figure of a mammalian tooth that most of you are very familiar with, of course. There's enamel, there's dentine, there's a pulp cavity in the center. If you've been to the dentist and you're older and you get drilled in there, it hurts because there's a nerve in here. Shark scale is exactly the same. There's an enamel coating, there's a dentine layer, and there's a pulp cavity, just like your tooth. And in evolutionary sense, if you trace this back through evolutionary time, really that's where your teeth came from. The genes that make these scales in sharks are very similar in patterning the basic mammalian tooth structure. Shark skin is interesting for a number of reasons, not just to biologists like myself who are interested in how sharks swim and how animals move in the water, but shark skin has generally inspired the fast skin wetsuits, the Olympic shark skin-like wetsuits that have so improved swimming speeds. They've also inspired these roughened ridge-like materials called riblets that people are now putting on wind turbines because it's thought that by imitating the surface of a shark even loosely, maybe the shark skin-like surfaces actually act to reduce the effort it takes to move through the water. It reduces drag, and that enables the shark to swim more efficiently through the water, might enable you to swim faster. It might reduce the energy required to spin the turbines and enable more efficient extraction of energy from turbines. So there's a whole field of sort of shark skin biomimetics, it's called, where we use information from biology to try and understand, try and improve human structures. So I was pretty interested in this. I wanted to know if the shark skin really worked. I wanted to know if the wetsuit material made by Speedo really worked. So how do we study this kind of thing? Well, one way to do it is to make little bending pieces of shark skin and swim them in my lab powered by a robot. So here's a dead shark. We've chopped out a piece of the shark. We remove all the skin and connective tissue and wind up with these strips of shark skin, which we know we have marked for orientation. We can then take those two strips and make a sandwich, a shark skin sandwich. That's very appealing to you. But it's a shark skin sandwich, and we can swim that in a robotic device. And what I'll show you here is this dark square is a piece of shark skin. It's swimming in a current. It's powered by a robot, which moves this vertical rod. And we shine some laser light at it so we can image the water motion over the shark's skin. So we can get a very detailed picture of how the skin moves, and we can measure how fast it swims. So the robot will move it back and forth. The shark skin wants to swim forward, and we can measure that speed. So here's a movie that wasn't playing all that well earlier, but let's give it a try, of the shark skin swimming in my lab, if you like. And this dark curved piece is the shark skin. And I show this also to illustrate to you a new technique that we've been using in recent years to study how fish swim and how fish robots and robotic devices swim. the image that you see is this speckly pattern here, which is laser light reflecting off of small particles in the water. So if I back up for a second, you see the laser light here. We've added a lot of small, very small reflective silver beads to the water, and this blue laser light is going to be reflected into a camera. And that's what we see. And these particles will deform and move as the robot moves, as the fish moves, or in this case, as this piece of shark skin moves. So let's see what this looks like. It's pretty slow, but you can see when the shark skin is going to bend down. And can you see, I hope, the water sort of moving down as the particles move down? You can see particles moving here and forming a circular little vortex at the front as it spins around. Shark skin is moving up, and here's the spinning region of fluid. This technique allows you to see water flow around swimming fishes and objects, which is tremendously valuable for those of us interested in understanding the mechanisms of propulsion. So let me just give you a quick result. I won't show much data here, but a couple of slides that might be kind of interesting, which I've simplified. So here's our shark skin, and all we're going to do is measure the swimming speed. And my point is just that when the shark skin has its denticles, little scales, the tooth-like scales, intact, it swims faster than if you remove them. So on the solid green bar is the shark skin surface sanded down, so we took off the denticles. On the left, they're intact, and it swims faster. It swims about 12% faster. And that actually is a lot when it comes to thinking about the fluid motion and how much energy it takes to move through the fluid. So the shark skin surface does very much seem to improve propulsion. Now, let's look at one of the imitators of shark skin, the Speedo wetsuits. So here's a sample of the Speedo wetsuit material, what they call the Fast Skin 2. Swimmers wearing these wetsuits, which are basically whole body sausage casings that they squeeze themselves into, did show very much improved swimming times. And so we were interested in knowing whether it was due to the structure of the material. You can see they've printed that with these fancy shark skin-like denticle patterns, very loosely interpreted. They did show very much faster swimming times. We wanted to know, is it the surface structure, or is it the fact that they've squeezed themselves into this, which just tightens their body, it improves blood flow back to the heart, it reduces their cross-sectional area. There are lots of reasons to think that it reduces bumps and wriggles on the surface. There are lots of reasons to think that this might work, but it's not actually due to the surface. So we imaged the surface of this special shark skin material, and to me this looks like fabric. Fabric with some dents that have been imprinted onto it, and if you look at a cross-section, you can see these dents are here. To me, I hope to you too, this doesn't look like shark skin. And interestingly, when you make a flexible membrane from the Speedo fabric, you swim it, you find out it either does nothing when you compare it to a control, or it actually swims worse when you compare it to a control. So from this, I proposed the notion that the improved swimming times that we've all probably heard about from swimmers wearing these wetsuits has nothing to do with the, quote, shark skin, excuse the air quotes, like function of the surface. It has to do with the fact that swimmers are squeezing themselves into these wetsuits, reducing their cross-sectional area, reducing the ripples from the skin, and other functions. So this is one sort of type of study. As an example, I'm trying to just illustrate for you how study of biology of sharks and the skin and the denticles on the skin can lead us to think about better ways we might design swimming suits. If you could make a wetsuit with real shark skin denticles on the surface, those little tiny 130 micron structures, I think you'd go much faster. But that's hard to do. Now let's move away from the cartilaginous fishes and my one example of locomotion and theme from the gallery upstairs to rayfin fishes. And this is where the vast diversity of fishes is shown. There are 30-odd thousand species now of rayfin fishes, a couple of hundred new ones described every year. I just want to say a few words about rayfin fishes and their grand diversity. So the rayfin fish, well, what is a rayfin? Sharks have fins, and they have rays in their fins, but they're not like these. Here's a bluegill sunfish. Here's the fin. It's sticking out its fin, looking at us through the aquarium in my lab. And you'll notice that the fin has these rays that come out and support it, and there's a very thin membrane in between, like a cellophane collagen membrane. You can think of this being actually very similar to your hand, but with joints and a membrane in between. But there's more to the rayfin fish, rayfin, than that. Let's look in a little bit of detail. If we have our schematic fish here with a blue box where the fin is, you can see from the big red image here, we've stained the bone red. So those fin rays that you see here are made of bone, and they're segmented. So you see little tiny segments here. If we blow up a segment, you can see these little tiny bony segments. Here's this brown area, brownish area here is the membrane that connects the fin rays. And not only that, but they're paired. They're divided into halves, a left half and a right half, a half ray. They're attached at the end. They come down, and muscles move them at the base so the fish can move its fin around. So each fin ray here is paired. There are two parts to it. You can't see it here, but it's paired with two parts. And that design enables the fish to do something very clever. If you pull on one part and not the other, it will curve and bend. And I have a very, some of you have seen this, very low-tech illustration of this. But it has an important, it's very important for the locomotion of fish. It's what makes the rayfin fishes, gives them their name. So I've taken the top of a Ziploc bag. I've chopped it off. I cut one end so you can separate the two zippy pieces here. And just like the fin ray of a fish, we have our two Ziploc pieces. We'll zip this together, and then my hand will play the role of the muscles at the base, and it enables the fish to curve the fin like so. This is, it amuses me anyway. I like to look at it. Now, what this enables fish to do, which, say, insects can't do with their keratinous wings, is it enables fish to apply force to a fluid as the fluid tries to flow past it. So normally when fluid moves down past a structure, it'll bend over like so. And so the structure will deform as the fluid moves past it. But a fish can curve the structure into the oncoming flow and resist that fluid motion. And no one can really say for sure if the 30,000 species of rayfin fishes, if the reason they're so diverse is because they have clever fin control mechanisms, but it's certainly a key feature of their morphology, a key feature of their design, of their functional design, that enables them to manipulate water and live in the three-dimensional fluid environment. So that's what a rayfin fish is. If you ever see rayfin fish, think Ziploc bag. That's the rayfin. Now, another amazing feature of rayfin fish is their prey capture system. And many of us here at Harvard have studied this on and off over the years. Here's a Goliath fish from the Congo. I think this actually was featured on that River Monsters television show. You can see it's pretty ferocious looking. And when you go upstairs and look at the fish gallery, take some time to look at the different kinds of jaws that fishes have. This fish looks like if it bit you, you'd be in big trouble. But another major way that fish capture prey is to suck the prey into their mouth. I've got two different movies here to show you what this looks like. This is known as suction feeding in fishes. So here's a poor little prey item. The next movie is going to be a little worse than this one. And here's the predator coming toward the poor little prey item. And fish jaws are known for being highly mobile, and more advanced fishes have highly mobile, extensible jaws that they can shoot out at the prey. And here's an example. As you'll see the predator kind of approach, shoot its jaw out, and suck that prey fish into the mouth. And this is a tremendously versatile way of capturing prey. This is something that Professor Lim studied for many years. You can capture almost anything by suction feeding. Some sharks and rays will use suction feeding, and sometimes they're camouflaged, so poor innocent fish swimming lazily across the bottom don't see the camouflaged shark, which can devour it very rapidly. There's not much escape from that. It's a really a rather dramatic and horrifying video, but that's suction feeding. And of course, deep-sea fish have well-known, very large teeth and big jaws relative to their head size for capturing what are probably pretty scarce prey items when you encounter one you don't want to let it go. But there are no high-speed movies such as you just saw of deep-sea fish feeding. And so biologists have resorted to sophisticated guesswork. We're trying to build some robot models of the deep-sea fish jaws now so we can begin to understand how they might have worked. But until we can capture these, bring them up, and have them feed, we really won't know exactly how the feeding system works. And there's some, in the exhibit upstairs, there's a whole section devoted to deep-sea fishes and life in the deep sea. Now, another feature of the exhibit upstairs is to highlight certain aspects of fish migration and ecology. And so let me say a few words about migratory behaviors in fishes. So here's a moray eel, and the eel family, the eel group, I should say, has some amazing features. And one of the amazing features, one of the two I'll talk about, is the migration habits of eels. So some of you may well know about this, but eels are born, if you like, in the Sargasso Sea area. The American and European eel species are born in the Sargasso Sea area. And they slightly different regions, and they migrate respectively to the shores of North America and Europe. And after that migration through saltwater, they enter freshwater, swim up rivers and streams, and live in rivers and streams along the coast for 10 to 30 years sometimes before they migrate back downstream, swim across to the Sargasso Sea, spawn, and produce the next generation of eels. And it's thought that this rather strange pattern arose from continental drift, that when North America and Europe were much closer together, there was a common population that diverged as the continents drifted apart. So that's one, just the migration of this and the ability of eels to swim across half of the Atlantic Ocean and then back again is amazing enough. But what's also amazing is that after the eels are born, the larval stage that they have, and that does that swim from the Sargasso to either Europe or North America, is a very strange thing. From insects, you certainly know that the larval forms can look very different than the adults. Well, it's true sometimes of fish, too. And the eels have a larva. Eels and their relatives, like tarpon, have a larva that's known as a leptocephalus larva, which means small head. And if you look at, this is the head, but that's what the head really looks like. It's a very small head. And they've got this big leaf-like ribbon-shaped body called the leptocephalus larva. And this is what migrates from the Sargasso Sea. When you see an eel in local streams, that eel started life as a leptocephalus in the Sargasso Sea, and it swam here and then metamorphosed into what looks more like a normal eel, which you can see here, where you start with a leptocephalus larva up here with a tiny head, and you see a slow metamorphosis here to a much more eel-like creature on the bottom. Now, there was recent video of leptocephalus larva swimming, and it was pretty amazing. This is in the Indo-Pacific region. And so this is a leptocephalus larva that's come back to the reef. It's going to transform into a moray eel and settle down on the bottom. And you can see the tiny head here. But it's an amazing ribbon-like locomotion style that it uses to swim. And they migrate thousands of miles swimming like this to get to a freshwater stream. If it's a moray eel, it's going to settle on the reef, metamorphose, but for freshwater eels, they have leptocephali also. So I want to transition now to the last theme. We've talked about locomotion a little bit with eels swimming. Now I want to transition to really the last topic to cover here tonight, and that is some of the new research that's happening on fish robotics. How fish swim and how they move through a very dense and viscous medium like water is of interest to lots of people. Every day in the paper now, practically, you see something about some robot somewhere doing something. Humanoid robots serving tea. You see snake robots. You see terrestrial robots, flying robots. There's a very good group here working on flying robots. Professor Bewiner, who kindly introduced me, his lab has done work on terrestrial locomotion and robotics. And there are those of us, myself included, who work in the water environment and who want to help understand aquatic robots, how we might use principles from fish to help design better robots. I'll show you just a couple of movies from some older work, not my own. This is from Japan. It's a sort of a swimming manta-like robot here. They did quite a nice job, actually, actuating the wings and having that robot swim. I think we're still very much, I should emphasize, at the early stages of fish robotics. So there's a lot of good work to do in the future. Here's a very famous robotic fish from the University of Essex, some very good engineers. This was originally displayed at the London Aquarium with a probably somewhat facetious notion that it would replace a real fish, and people would visit the aquarium and see the robot fish swimming around. But they did a nice job. And so here's the robot fish. It looks, the innards are shown up there in the upper left. But you can see they've got the motion quite well. There are some sensors there, so it can turn. They did an excellent job, actually. And in the exhibit upstairs, there are some robot parts you can see and a prototype whole fish robot. So here's one collaborative work that my lab has been doing with Drexel University and my very close colleague, Professor James Tangora. This is modeled on a bluegill sunfish, the one I used to show you what a Rayfin fin is like. These are sort of simplified fin rays here, but each one of these has motors that control the motion of them. The body is stiff, but it has positions for sensors, so we can sense the water flow. The prototype of this is upstairs. When you pull it apart, you can see some of the controls for the different fins. And this robot is now undergoing some final testing, waterproofing. One of the reasons I think fish robotics is a little bit behind other areas is that when your robot leaks, it's very bad. So it makes it a little harder. It's a lot harder to do things when you have catastrophic failures. It can set you back a long time. But James's lab has done a fantastic job designing all the actuators, and this fish can move its fins. We're now working on making it more robust and controlling it so we can tell it to do what we want. But I think this is the first whole fish robot with actuated individual fins. Other robot devices are used, and we've used, to help understand something about what fish do in a way you really can't do with a live fish. So robotics, rather than just building cool devices that might be useful, they can help you understand biology. It's an approach to biology also. So here's an example. I'm going to show you a movie of this fish, which is a ghost knifefish, swimming. And this fish uses its anal fin, which is this long ribbon-like fin here, to swim. And you can see the waves that pass up and down the fin. It's a rather strange-looking fish. Comes from the Amazon. It navigates in dark waters. Actually has an electric sense, so it can sense electric fields. But you can see the control that it has over that wave on the fin. And each one of these little lines is a fin ray. So it has more than 100, roughly 150 individual fin rays. Each one actually has 4 muscles attached to it. So there's just a tremendous sophistication of control. So we can study the fish, but it's really hard to control the fish. The fish kind of does what it wants. And we'd like to be able to understand what these patterns of wave-like motion mean. Can we use those for a robot? If we can make wave-like motions, what can we learn from the fish? So collaborative work with my lab and Northwestern University has allowed us to build a robotic model, not a robot fish, but just a model that has some of the key features. Was still very challenging to do, that we can use to explore the wave. So one thing, for example, you can see, if you look at that wave carefully, the fish will go forward, it'll go backward, and when it goes forward, as you might expect, the wave on the fin passes back, which pushes water back, which makes the fish go forward. If the fish goes backward, the wave on the fin goes forward, which pushes the water in the right direction, so the fish goes backward. But there are a few moments where you'll see the wave come together in the center and collide, right there, for example, and the fish goes up when it does that. So we built a robot. Here's the knifefish robot. Granted, it's a pretty abstract knifefish robot, but getting 32 actuators with each one of these being a fin ray to work and not leak, it was very difficult. And we can make this robot do more or less what we want. So we can make it wave, we can pass waves of bending down the fin, we can pass waves forward, we can make colliding waves, we can change the frequency, the amplitude, we can control everything. And that's a fantastic thing for a biologist to be able to do, because fish are notoriously hard to control. So I love robotics because I can control things, and we were able to show that making a colliding wave actually pushes the robot up. But something we could observe in the fish, but we didn't know if it was causal. We can also make very sort of simplified versions of fish tails and flap them in the lab. And we can then test our mechanisms. We can test, can we control this? We can test the strength of the connections. Do cables break? Do we have problems? can use then to then incorporate things like this into the whole fish robot. So this was a project on the tail that we used to do, and this, what we learned from this went into that whole fish robot that you just saw. And we can control this tail motion quite accurately and make it do things, make the shape of the tail do things that fish tails do, which is quite complicated. Many fish robots you'll see just have a flat plexiglass plate that kind of flaps back and forth, and that's a little sad because fish are much more complicated. So we've tried to go beyond that, and so we have tendons and things that come down through the body of the robot, come back, attached to a tail. We print parts of the robot with 3D printers so we can make them of the stiffness we want, the shape we want. We can alter that, and we can make the tail do things. So you're looking from behind now just to see the tail flap up and down in a very simple way. We can make the tail roll back and forth, make the tail kind of cup. So if you look at this lower left-hand panel, you'll see the edges move faster than the center and has a cup-like shape. And this is actually something that's very important that fish do. They make these cup-like shapes with their fin, much like you might do if you're doing the freestyle and you're cupping your hand into the flow. And we can measure the forces that each of these motions generates. So robotics has been a tremendous tool, and I think will continue to be, to allow us to investigate different patterns of motion and what force they produce, to design vehicles that will do things we can't do now, and to better understand how fish are designed, and to test things, test hypotheses, and answer questions we can't answer with live fish. So let me wrap up and conclude and just say that the gallery that you'll see when you go up there, either after today or in the coming weeks, and I certainly encourage you to bring family, friends, children, and so forth up. The whole museum is fantastic, but the new fish gallery we're especially happy about. And explore some of the aspects of the diversity of fishes. Fish research at Harvard has been here for a long time. It's been a very important part of the biology department, of the Department of Organismic and Evolutionary Biology. Been a long series of important characters who have contributed, not the least of which was Professor Lim here. Urge you to look at that panel on the end of some of Carl's contributions. And I think that overall it's a very exciting time for research on fish biology. New species are being discovered all the time. There are all sorts of new approaches being taken to studying fish with micro-CT scanning so we can see the anatomy inside, robotics, biodiversity, new collections, new species are being found constantly. It's a great, wonderful world of fish out there. And I hope you'll come see the exhibit and at least indirectly participate. So thank you very much. So I'm happy to take questions, if I can. Yes, right here.
Robotics, have you been approached by military or intelligence people for applications?
The question is, have I been approached by military or intelligence people for applications? And the answer is yes, but it's not as sophisticated or as nefarious as you might think. So yes, the Navy does provide some funding for our robotics work. We don't do classified research. It's all published. It's all out there for anybody to read and see. Like many organizations, the Navy is also interested in exploration, in being able to find things, being able to move quietly through the water. You can imagine various applications for that, I'm sure. But there's just a basic level of technology that organizations like the Navy fund that help us better do things as a society, building robots. There's lots of research funded by the Department of Defense on snake robots, and one of the applications of that is in earthquake areas where there's lots of rubble. You can send those snake robots, hopefully in the future, through that rubble and find people and listen for people. So the answer is yes, and I've actually been very pleased at how our interactions with the Navy have gone. Yeah. That's a very good question. The question is about the swim bladder function of fishes, and due primarily to time constraints, I kind of ignored the swim bladder function of fishes, but it is a really key feature. So one of the problems of moving in a fluid environment is controlling your buoyancy, controlling your body position. There's only small gravitational forces that you're forced to encounter because your density is slightly higher than that of the water. So fish have swim bladders. Not every fish, but most fish have swim bladders that can, most rayfin fishes have swim bladders, filled with gas, and it enables them to counteract their dense bones. Sharks, and we do talk about this a little bit in the exhibit, sharks many shark species have a very fatty liver that has a low density oil in it, which also serves an analogous function to the swim bladder. But swim bladders of fishes and our lungs share a common evolutionary history. And the exhibit will tell you a little bit about the two major designs of swim bladders. It's actually a fascinating topic because of how fish are able to get gas into their swim bladder even when they're down at very deep pressures. So there's a whole tricky physiological mechanism that enables fish that live 1,000 feet down to fill their swim bladder with gas. I'd be happy to talk to you more. It's a tremendous innovation, I think, that fishes have, that it really is opened up the three-dimensional aquatic habitat to them. I'll get to you guys over here. Go ahead.
When you and your students were out in the Charles, what did you find, and how was it different from 1790 or something?
Well, that's a very good question. So when we were out collecting in the Charles, what did we find, and how was it different than 1790? 1790 might be a little early, but the Charles was horribly polluted. There were various paint factories, which paint had a lot of lead in it back then. So there's a lot of lead and sediments in the Charles. I don't know, in 1790, I'm not sure what the water quality was, but certainly in the 1800s, it went downhill very rapidly. And there was not a lot of fish life in the Charles probably around 1900. We found, actually some eels in my lab right now that came out of the Charles River. So we found eels, you find bass, you can find some sunfishes of various kinds, herring various times of year. There's not a big diversity, but it's not too bad. Some catastomids, some suckers. If you know what a sucker is, it's a minnow-like fish. Some pretty good sized fish. Some small bass occasionally. We've been collecting for years in the Charles, and there's a nice little diversity. It's very close to Harvard, so that works well. I think the Charles is much better than it was, and certainly in the 1800s. And I think what you lose now that you may have had still back then is some of the migratory fishes because of the dams that have been up by Watertown. There's a dam, and other dams. The locks down by the Museum of Science are really, really not good for fish. But it wasn't that long ago, I think someone saw a sturgeon going through the locks in the Charles River. The Globe had a picture of it. So there's some return, I think. Yeah?
Are the denticles the same on male and female sharks?
Generally, yes. There's not a big sexual dimorphism. They vary a lot on different parts of the body. So that's where most of the variation in one species of shark would be. They're pretty similar. I don't know anybody that's looked in detail at that, but my first rough answer to your question would be that they're pretty much the same.
I would like to ask if you could comment on the frequency and the evolution of fish, the relative number of fish that have come from land animals that have gone into and adapted to, and whether or not this is a big component of the life variation in fish now.
So there are some fish that have emerged onto land. And they don't live permanently on land, but they can move around on land. Even eels move quite well across damp ground, if that's what you mean. Or are you thinking more broadly with invertebrates, so mammals that have so certainly it's one of the greatest cases, I think, of convergent evolution, where environmental pressures dictate very closely the design of animals. And you see that when mammals that used to live on land, like whales are a good example, their ancestors are land mammals, re-evolved to live in the water, they assume fish-like shapes. Obviously, whales, porpoises look a lot like a tuna. And that's one of the key examples of convergent evolution. It's one of the reasons we believe that natural selection, where you have a very costly behavior, such as moving in the water, which is very hard to do. It's very energetically expensive. Places very strict demands on the shapes you can take, and so we see that convergence when multiple groups come back into the water. So that's, I think, the main lesson from that type of re-entry of the water in mammals. Fish themselves don't really do that. There are many fish that come out onto land. There are fish that breathe air quite extensively. I haven't talked about that much here, but there aren't many that really have evolved a fully terrestrial existence. Really, all fish reproduce in the water for the most part, with a few strange exceptions. And I think that's the best answer I can give you now. Oh, yes?
Have you ever considered using fish robots for exploration in the ocean?
Absolutely. So the question, it's a good one, is have we ever considered using fish robots for exploration in the ocean? Absolutely. That's our long-term goal. It's just, it's hard to do. And building the robots is hard to do. So it's going to be a while. I'll probably be retired before we get really good at that. But that's absolutely our goal, is to have quiet, fin robots, robots that can bend, robots that can move their fins. They move, they maneuver quietly. And you can explore, you can sample water, you can observe coral reef fishes in a way you just can't do with modern propeller-driven robots. So absolutely, that's our goal. But you're going to be the one to figure that out, probably not me. Yes?
There's a lot sometimes in the news about how bigger fish species like salmon, cod, they're facing serious stresses on their populations. And I guess since you have a very long-term perspective on fish, you can speak to any other time periods where fish have faced or certain populations, some of the bigger populations, have faced similar serious stresses.
Boy, that's a good question. It's a tough question. I don't have a great answer. There's no doubt that fish populations are under massive threat now. I think the only thing I could say through time, it would be to go way back in time, to with continents are drifting and major changes in habitats are occurring, such as shore habitats are being destroyed by collision of continents, for example. And that certainly has happened and radically changed the habitat available for fishes. I don't actually know of anybody who's tried to estimate the effect of that on fish populations. But that's actually the closest thing, other than if we think of meteor impacts or things really changing the atmospheric conditions that very much influence the ocean. No one that I know of has actually tried to make a reasonably good estimate of what happened to fish populations at that time. We all think of dinosaurs going extinct because meteors hit the Earth. But the fish were certainly very much affected. But I think nobody really has a good idea how much. But certainly, I think what's happening in modern times is probably far worse on a global scale than even those catastrophic events in Earth history. Yeah.
Are there data on the psychological needs of fish?
On the psychological
needs of fish.
Are there data on the psychological needs of fish? Well,
Think about the
fish are,
fish, etc.
I think it's a very interesting question. Fish have personalities. Some fish seem smarter than others. Some fish seem, more wily than others. And there's been a lot of research on the sensory lives of fishes. How do they sense their environment? I didn't really address that tonight. But how do they sense currents? How do they sense pain? They sense taste. And that world of fish is pretty well understood. In terms of behavior and the social interactions of fishes have been worked on a great deal and continue to be for various reasons. Brain chemistry changes, hormonal effects on brains have been studied quite extensively on fish, and it's been influential in modern medicine, actually. But I think in terms of the broader psychological life of a fish, it's not that much is known, actually, but it's not because it's not important. I think it's just hard to think about how to study that. It's hard to be inside the fish brain. We try in my lab to give them environments that they seem to like, but it's not, I think, much is known. Yes?
How do schools of fish execute their movements so perfectly and quickly? Is there a feeder?
This question is about schooling fish. How do they school and how do they execute their beautiful rhythmic motions? That's a topic that's under a lot of study right now. There is some evidence that there can be leaders. In fact, one of the Harvard students who took my fish class a few years ago did some work in Peru this last year and showed that there seemed to be some leader fish or sentinel fish, which kind of are monitoring the environment, and other fish will watch that fish and react accordingly. Typically, the big, beautiful images you see on the Discovery Channel or Planet Earth where schools of fish are moving in unison and so forth. Fish have very exquisite ability to sense water flow over the environment and to sense pressure changes near them. So they can sense where neighbors are in a big school and adjust their their behavior accordingly. And they're also very visual. So when they see leaders start to turn or part of the school start to turn, they may turn with them. There's been a lot of work done. It's not all very conclusive that schooling might reduce the energy it takes for fish to move through the water. I don't actually subscribe to that very much, but it certainly makes it more difficult for predators to attack them. So it's thought that a primary function of fish schools is anti-predator, and the fact that the schools are sort of mixing and fish are changing position. Typically, there's not just one leader all the time, but the ones from the back will go forward, and there's this big, beautiful mixing of the school. Makes it very hard for predators to pick out individual fish. They're obviously somewhat successful, but it does increase the difficulty of it. So I think the primary answer to your question is that they sense patterns of water flow and pressure change, and they use visual cues to orient themselves with respect to their neighbors. It happens, it's very quick, it's beautiful, but if you stimulate, produce a pressure wave in a tank of fish, many of them will do the same thing very similarly. So they just have very exquisite sensory capabilities. Yes?
From your research, how many oceans have you studied, visited, and of those bodies of water, which one had the most interesting quantity or variety of fish?
Oh, I've been a lot of different places. The question is, of the oceans I've been to and studied, I don't do a lot of field work, but I've done a lot of scuba diving in various oceans around the world. And so where did I find the most interesting? I spent a lot of my youth scuba diving in the Mediterranean, actually, as an underage diver that probably wouldn't be allowed now, but influenced by Jacques Cousteau, going through the underwater world. And the Mediterranean is a fascinating area to me. I mean, I've been diving in Hawaii, I've been diving in the Caribbean, and I think off the West Coast. I think the Mediterranean is an amazing area. I don't know if many of you know this, but the Mediterranean is quite recent, and it actually completely dried up. Now, how many years ago, I've forgotten, but a number of million years ago, it was completely dry. So as you drill through the Mediterranean and go into the bottom, you have these huge salt beds where the salt water had evaporated and dried, and then it would flooded through the Straits of Gibraltar. So the whole fish fauna is different than you might expect. It's, that would be my choice, I think.
Yes, most fish propel themselves from side to side, but I noticed that you had a model going up and down, 9 million. What percentage of, if any, fish actually propel themselves?
It's a good question. So my fish robot that you saw was just, imagine it's like a fish on its side. And really, almost all fish with their backbones propel themselves by moving their tails from side to side in this way. Whereas it's a nice example of convergent evolution when land mammals came back to the ocean. Of course, porpoises and whales move their vertebral column up and down. So you can see they're streamlined, but you can see the relics of their terrestrial heritage and how they move their tails. The robot was designed that way purely for convenience, for our ability to study it. It was a lot better for us to have it on the side for our imaging, force measurement purposes. So that's really the only significance of
I
that.
had read that they found a fish that actually, it's a fish that goes top to bottom instead of side to side. I'm not
Okay, maybe I'd be interested in knowing more about that, actually. I don't want to ignore you guys at the back.
This is about the leptocephalus larvae. How do they know what they're going through the migration?
Oh, boy. That's a tough question. So how do the leptocephalus larvae know where the heck they're going? I added the where the heck part. Really, I have no idea. The only thing I can say is there's a lot of research now that's fascinating on magnetic navigation. And they're finding small magnetite mineral particles or neurons that are sensitive to magnetic fields. So it is possible, like turtle navigation has been worked on quite a bit, the big ocean-going turtles, and how they navigate probably is by magnetic sense, by following the Earth's magnetic lines. And leptocephalus larvae may use some combination of magnetic sense, ultraviolet light, like polarization, instinct, pre-programming from ancestors. I really have no good answer to that question, but it'd be a great one for you to study. There are all sorts of fascinating topics, and that's a fantastic one. And their brain is tiny. I mean, there's not much there.
Are there other?
One more question. I apologize. I'm happy to stay for a little bit and answer more questions. Yes?
Are there other catastrophe species other than eels?
there are a number, there's a whole, in fact, the exhibit talks about some of them. There are a number of fish that are born in the ocean and come into land to breed. Yeah, and the anadromous reverse of that, where you have fish that are born in streams, salmon are a good example, and then move out to the ocean for adulthood and then come back. So there are many different, pretty much every combination in between you can think of, or fish migrating from one habitat to another. It's a very important part of fish life histories, and I could give you other examples. We can talk more about that. That's one reason we highlighted migration actually in the exhibit, is that the ability of fish to move. And through habitats, they're changing their physiology when they do that. And of course, when we build dams and do things that inhibit that migratory route, it's deadly for the fish. So thank you very much. So I would be happy to take a few more questions, but I know we have the museum members, we have to head upstairs, I think, soon. Thank you for your interest.